So I'm so glad to be here. I actually founded the Multiple Myeloma Research Foundation back in 1998. I'm sure many of you are familiar with the disease, but multiple myeloma is a blood cancer in the same family as leukemia and lymphoma. But unfortunately, when I was diagnosed with the disease at 37, the average lifespan of a myeloma patient was just 3 years, and it was uniformly fatal. It is still considered to be uniformly fatal today, but we've made tremendous progress. The way we started the Multiple Myeloma Research Foundation was not looking like your typical advocacy group. And, you know, a lot of the advocacy groups look a lot different today, but you have to remember this was 1998 that we were building this foundation. So the way that we started to look at it was we wanted to attract as many scientists to the field as we could in myeloma. We wanted to get everybody working together as quickly as we could. And we wanted to get industry to support us as well. So over the years, we have built a model that's pretty much an end-to-end solution now in precision medicine. And I'll walk you through how it functions just quickly, and then we can turn it over to the other members of the group. And then I'll come back and tell you where I still see flaws in our system, especially in terms of where, when, how we can share data well. So if you look at our data bank, which is also a tissue bank, we asked many of the academic centers in myeloma to start banking tissue centrally and providing their tissue to a central bank that we were spearheading and running. As we did that, our early tissue that we were garnering back in 2003 and 2004, so ages ago, was really one-off tissue. It was not longitudinal tissue. It was kind of taking all comers into the tissue bank that we could get. But along with that, we then reached out to Eric Lander at the Broad Institute. We were both serving on the National Cancer Advisory Board together at the NIH, and said, what if we start sequencing some of this, these genomes in multiple myeloma? So we started doing that, and indeed started to make progress on the genomic sequencing side. But the myeloma team was still hugely frustrated that we didn't have that beautiful genotypic and phenotypic data that was longitudinal, following myeloma patients, not just from that early moment of diagnosis before they've been treated, but also through relapse. So we also fielded a study called the COMPASS study, and it's kind of sad that a research foundation had to take on building a $1,040 million patient study, but we wanted our data to go into the public domain, and we knew if we didn't do it as a trusted third party, it was going to be really hard, especially when we were starting this years ago, to get academic centers and industry just to turn over their data to a third party like us and say, well, everybody go share it. So we now have different gateways that are holding a lot of genotypic, phenotypic data in multiple myeloma, and then we're also building a registry direct to patient. So that's the left-hand side on the data bank. Move quickly to the Learning Center, which is where we push all of our data out to the public domain. We worked with a company called Genospace to build our gateway and put all the tools so any researcher can go on, look at our 1,000 patient study as well as all of our genomic information. And then we have companies like GNS up here in Boston that can literally take our raw data and put the myeloma pathways together and serve all the modeling support that our academic centers and industry need. And we literally host data jamborees up here in Boston and invite all the academic centers and industry to look and see what we're doing with GNS. Move forward to the clinical side, we have our own 22-member clinical network. And we do phase 1 and phase 2 trials. Within that clinical network, we've spearheaded about 60 trials of about 30 different agents. Now in myeloma, 7 of those agents have been approved, 3 more in phase 3 testing, and myeloma patients live much longer than the 3 years than I was diagnosed. It's approaching 9 or 10. And the patients are getting much, much more involved. And just to give you one example of the progress we made is early on with that early genomic study, we did identify BRAF as a target in myeloma. It's actually a melanoma target. We used our Learning Center to know that BRAF was interesting in myeloma, and there were some good melanoma drugs, but we also found through the Learning Center that MEK was equally as important, and we'll be launching a BRAF-MEK trial this year, and identifying the 6 to 8% of patients that have that target in myeloma through our own clinical network. So I'll stop there because there's still challenges in the system, but this is what we've been doing for the last 15 years or so.